Hello? Hello, can you hear me? Hello? Hello? No, sure.
We can hear you.
Oh, okay.
Would you like to set up your screen to share?
Yes. Can you see the screen?
Yep, yep.
Okay. Let me know when should I start.
I think we can start and then I'll just let in the rest who's coming in.
Well, let me turn on my video camera. Shall I start right now?
Yeah.
Okay. I will give an overview of regulatory trial design and application of complex innovative designs for gene therapy and the rare disease drug development. And this is an overview. Followed by that, there is many talks relating to these topics of complex innovative designs. And they are all non-technical and geared for general audience. Here is a brief outline. I shall start with challenges in rare disease drug development, and then look into legal and regulatory pathway for innovation. Then get into high-level overview of complex innovative designs, and then give a few examples of applications. This is a cartoon that summarizes lots of things about rare disease. First of all, 1 in 10 Americans have rare diseases. That means actually, it's pretty common, in fact, but then there's about 7,000 rare diseases, and then about 650 don't even have a treatment. So lack of treatment is a big problem. That's why there's so many innovative companies are engaged in development of treatment for rare diseases. Now, part of the difficulty is not only it's a small sample size, but also there is a lack of knowledge on the disease. That's why it can actually take a really, really long time for a patient to be diagnosed with a particular rare disease. And then even though for a particular rare disease, even though a patient's actually figure out he or she has a particular rare disease, and then by the time patient's diagnosed, and then the patient will find out there's not a whole lot of research done by the rare disease, and therefore it makes so many things very difficult in terms of drug development and also clinical trial designs. But then, again, if we talk about a single rare disease, usually there's not a whole lot of patients, and an ultra rare disease has even fewer patients. But then together, it represents a big population, and together, we can actually, work on things that are common, develop common tools so that different rare diseases can have a pathway for drug development. Moussiki, who is now at pharma, head of pharma, once famously said when he was at the FDA, that small population often restrict study design and replication and use of usual statistical inference. The problem of lack of research and knowledge clinical development is really serious. And first of all, we're talking about heterogeneous disease population. A particular rare disease have a particular way, particular path of progression, and then a patient cannot be diagnosed properly. You end up actually, a population of patients who are at different stages of progression, and that actually increases heterogeneity of the disease population. And then because of lack of research or lack of knowledge, there is numerous known, but then mostly unknown risk factors that also contribute heterogeneity. And then there is a lack of adequate natural history study of real world evidence. And then you have individual investigators or doctors who treat patients, but then people don't actually, get together, collect natural history data in a systematic way. And then to be honest, there is a lack of adequate number of research-oriented investigators who is capable actually doing this sort of things, clinical research. Well, that's actually a tremendous burden, for a rare disease, a drug development. On top of that, we have a regulatory burden. In fact, in 2009 February, FDA issued a draft guidance on rare disease. It pretty much says, well, regardless whether it's a common or rare disease, there is a single statutory requirement has to be met, which is a substantial evidence. And then, well, the law defines substantial evidence in a particular way. And then you have to deal with reviewer's bias on what it is substantial evidence. It is commonly interpreted as a randomized trial and a Euro statistical inference for substantial evidence evaluation. And then the problem is actually, that's on the regulatory side. On the industry side, there's a lack of innovation. Former FDA Commissioner Scott Gottlieb said several years ago, you wanted us to help modernize R&D. Now go out and do it. it says, unfortunately, we've seen a continued reluctance to adopt innovative approaches among sponsors and the clinical research organizations. In some cases, the business model adopted by clinical trial establishment just isn't compatible with the kind of positive but disruptive changes that certain innovations can enable. Well, I will give a very good example. Just yesterday, I think there is a company, I think in Boston, that actually announced that their drug development for Alzheimer, no, for ALS, Lou Gehrig's disease, failed. And then when I actually look at the press release, I was just, stunned that the problem that the company encountered is the same problem that another company encountered 24 years ago, which is a cephalon. When I was at FDA, I reviewed mytrophin for ALS. Now, the pattern of the problem is very similar, that patients are heterogeneous. There is a subgroup of patients that drug can be beneficial, yet the primary analysis and design focuses on the entire population. So you have a subgroup that doesn't really work, and then the subgroup, the drug doesn't work. So this kind of thing, lack of trial failure. And then, it's just actually incredible that, given so many years of research on adaptive designs and that aim to resolve this type of problem, companies in pharma still use old-fashioned design that simply doesn't work. Another example is Karyopharm selinexor for relapse refractory multiple myeloma. There is a single-arm trial, and then there is a natural history study. In February 2019, FDA ODAC concluded that the evidence generated from the analysis of RWD is not adequate to provide context or comparison for the overall survival observed in the STORM patients. Specifically, KTI original analysis, demonstrated there's a very huge treatment effect, the hazard ratio of 0.41 with very significant p-value. However, the problem is that they are comparing patients with relapsed refractory multiple myeloma with many times newly diagnosed patients. So it's not really a good comparison. When FDA started reviewing the application, FDA said, well, you got to actually, adjust for index state. You have to compare patients who start at who enter into the stage of relapse refractory multiple myeloma. After doing that, the p-value is barely significant. And then there's other selection criteria, for adjustment. And at the end of the day, the analysis yield a p-value of 0.33, and then the confidence interval 0.25 to 1.58. And then as a result, Karyopharm did not actually get approval based on survival. And instead, FDA figured a way to give an accelerated approval. So therefore, in order to overcome all these difficulties, it is important to go forward with innovative designs. Fortunately, there is a legal foundation and regular pathway for innovation. So it starts with substantial evidence, which means evidence consisting of adequate and well-controlled investigations. And then the 21st Century Cures Act recognized the importance to stick to substantial evidence criteria, but then recommend to use complex adaptive designs. And then it also recognized to use a real-world evidence as a basis of comparative analysis in new indication and post-approval studies. And then, there's all type of real world evidence, real world data. And the most importantly is from observational study and registry studies. And the law also gave FDA flexibility to use real world evidence for purposes not specified in the law. And then FDA did issue a framework of real world evidence for new indications, post-approval studies, and biological products. And then also recognize FDA has in fact used real world evidence in oncology and rare diseases. However, there are issues of using real world evidence in terms of systematic bias, missing data, especially in retrospective studies. And 2019, FDA issued draft guidance on natural history studies and point out 8 limitations of retrospective natural history studies, which include big 3 C's: comparability, contemporaneousness, completeness of data, and also all sorts of selection bias. It can be characterized as time length bias, publication bias, and enrollment criteria bias. In 2018, Curry, Sachs, and Woodcock wrote a paper in JAMA, which pretty much doubt the use of real-world evidence alone as a basis for comparative analysis, comparative effectiveness analysis, and emphasize the need for randomized controlled clinical trials. In this context, the framework, recognize RWE can have many uses, including improvement of efficiency of clinical trials. One example would be using RWE to form prior probability distribution in Bayesian statistical models. There are other ways to utilize real-world evidence. Doesn't have to be Bayesian, can be frequentist meta-analysis or even match-controlled analysis. And then the FDA recognize, you cannot avoid the bias. Given there is a bias, external control is most interpretable when treatment effect is large in comparison to potential biases and unknown variabilities. It also recognizes a hybrid approach of using external control data to add to concurrent control in a randomized trial. And then 21st Century Cure Act also state that in order to use a real-world evidence from natural history study, one has to prospectively plan and design observational studies and other analysis. And about 20 years ago, this is already recognized by SHE 10. It requires pre-specified analysis. And regarding method analysis, it has to use matching on selection criteria or adjustment made to account for population difference in the sense of using regression analysis. It also encourages to use multiple historical controls. This is important because one can never know what kind of bias is present in a single natural history study. So if multiple controls are available, that would certainly going to increase the robustness of a finding if a new drug is effective. And FDA also issued gene therapy guidance for rare diseases. It recognized a patient can repeatedly use in clinical development program from phase 1 all the way to phase 3 and post-approval studies. There are 3 basic designs. One is a parallel group design. The second one is a single-arm trial with natural history controls from initial observations. And then there's another type of single-arm trial with external natural history controls. Now, this actually provides the basis for the next section, which is complex innovative designs. And I'm going to cover 4 different designs. One is a single-arm trial with external natural history study. The second one is adaptive hybrid designs. The third one is RWE randomized enrichment design. The last one is randomized delay start design. The first type is really simple. You have a single arm trial, and then you have external natural history study of real world evidence. And then one can digitize from multiple natural history studies in the literature, or just extract, summary statistic. This would satisfy multiple historical control requirement. However, the analysis could be actually really, really complicated, sophisticated, or even or has to be very rigorous. It starts with exact matching of the index date, because natural history studies are collected over a long period of time. A patient enter a study when was first born, but then a particular study do not actually enroll just a patient. Maybe actually, patient passed 1 year, so you cannot actually start comparing, data with 1 year old. You have to identify the index date and then extract the data. And then once you've done that, you have to actually control other risk factors. And then there's a method of analysis can be really sophisticated. It's all designed to really help out to address the potential issues or limitations of natural history studies. And the methodology are developed for continuous variables, categorical variables, and time to event variables. The second part is adaptive hybrid designs. And then, clinical program consists of a phase 2 program of treatment from single arm trial design, and also a natural history of RWE of controls. And then after phase 2 or phase 2 trial, single arm trial, one enter into a phase 3 randomized control study where patients are randomized, for example, in 2-to-1 randomization ratio to treatment and then concurrent controls. And then results from both phase 2 and phase 3 can be combined in such a way that increase statistical power and increase robustness of statistical finding. This is all designed to really actually address the issue that it is very difficult to enroll enough patients in a given period of time and to power a randomized trial using traditional standard. The second adaptive hybrid design is a 3-stage design. Essentially, an interim analysis can be added to the phase 3 randomized control design. And then that add additional flexibility in terms of addressing some of the uncertainties in the trial design. The next one is randomized delay start design. It's a 2-stage design, but then it has a screening phase. Patients are screened. Those who qualify will be randomized to treatment and a control. In the second stage, patients receive control in the first stage are crossed over to treatment. Depending on the type of endpoint, if the endpoint is objective, there's just no need to do a randomized withdrawal of patients who were initially randomized to receive treatment. But if the endpoint is subjective, it is highly important to blind the study. It may be actually important to do some randomized withdrawal of patients who initially randomized to treatment. With a randomized delay start design, we're looking at delta 1, which is a traditional parallel group comparison, and then delta 2, which is actually intra-patient comparison. So you look at what's observed after patients switch to treatment compared to what's predicted had patients not received the new treatment. So this delta 1 and delta 2 both characterize efficacy, and then those can be combined to raise statistical power and reduce the sample size. And the key here is that we need to have a consistency of the result. You cannot have a parallel comparison give a negative result and then intra-patient comparison give a positive result. It has to be consistent so that it can be combined. The emphasis here is in incorporating intra-patient comparison, which by the way is most relevant to patients. Patient enter into trial, want to slow down the progression or improve from the baseline of their disease conditions. The last one is RWE randomized enrichment design. It starts with a screening, and then patient who qualify enter into stage 1, which is an open label observational study. At the end of stage 1, patient enter into a randomized control trial stage where patients who qualify will be randomized to receive treatment or RWE. Now, the analysis actually is very, very interesting. So we want to actually analyze the data using using actually delta 2. Delta 2 is actually an intra patient comparison of what's observed versus what projected. However, there are certain diseases or clinical endpoint that may be subject to placebo effect. So because of patients are randomized to receive treatment and control, one can characterize, what's the placebo effect. With this knowledge, one can actually make adjustment to delta 2 so that the result is robust. I think I started late. Can I actually just go through this really quickly? Hello?
Yep, sure.
Okay, so let's actually look at a few applications. The first one is a single arm trial with external natural history control used for breakthrough therapy designation for LOPD, which is a late onset Pompe disease. The second one is a single arm trial with external natural history control. It's oncology trial. And then at the time, this presentation was prepared a year and a half ago. I was actually in the process of developing a match control methodology. And then this one is adaptive hybrid design, and then is for rare lysosomal storage disease. It has a phase 2 and retrospective study. It uses a 3-stage adaptive design as I presented. And at the end of the first stage, there is sample size adjustment. And then there is a sequential testing for second and final stage. You use a exact statistical inference. The second one is a 2-stage adaptive design. So it's very similar in nature, but it's actually used for gene therapy. And this one is RWD, randomized delayed start design. It's submitted as a post-approval commitment as part of an NDA submission. So at the time, NDA is submitted based on biomarkers. This commitment trial is also submitted as because of using this design, the trial is 50 or 80% more efficient. And then the drug was approved, and FDA agreed in principle, but requested more simulation studies to really look into the operating characteristic of the design for that particular application. And then this one is RWE randomized enrichment design. And then the design lead to 50 to 60% of reduction of the sample size compared to parallel group design. But then the design was abandoned because of difficult to enroll, and also at the time, the FDA's guidance has now came out that there's a strong reviewer's bias that's actually prevented this design from going forward. And then here's another use of the same design is gene therapy for rare disease. And then this is actually, become very common nowadays to utilize this type of design. And the trial is going on, and then, blinded data are being monitored. That also lead to improvement of the statistical analysis. And last but not least, and clinical trial technology platform is needed to implement various design. There's also a need to develop analytical capabilities. One possibility is use Amazon AWS system. And one need to engage in adaptive statistical planning using blinded data. But then very importantly, need to work with specialty service provider that well-trained on methodology, technology, and the process, and also highly motivated, efficient, and productive. Thanks you for everyone. If we have time, I can answer a few questions.
Is there any question? I think in that case, we'll end the call, and we'll see everyone on the next presentation. Thank you, Ching.
Thank you. Bye-bye.
Thank you.